webinar hosted by the Canadian Biomaterials Society Ottawa Student Chapter. It's my privilege to introduce Dr. Elena Martinez. Dr. Martinez holds a PhD in physics from the University of Barcelona. After her postdoctoral studies at the EPFL and Imperial College, she settled at the Institute for Bioengineering of Catalonia, IBEC. During her scientific career, she has supervised 9 PhD students, 4 theses currently ongoing, published 106 papers, and participated in national and international projects. She has also established fruitful collaborations at the national and international levels. Dr. Martinez has been the leader of the Biomimetic Systems for Cell Engineering group at IBEC since 2018. Her group is currently composed of 14 people from a diverse scientific background. Current projects include in vitro modeling of drug-induced arrhythmia in cardiac engineered constructs and the in vitro modeling of intestinal tissue using adult stem cells derived from intestinal organoids for the study of intestinal development and disease. If you have any questions during the presentation, please send them through the Q&A to the chat. Dr. Martinez, thank you for joining us this morning, and we look forward to your remarks. I will pass the microphone to you.
Good morning, everyone. They are in Ottawa. It's in the afternoon here in Barcelona. And I would like to thank everyone, and especially the organizers, for inviting me. So it is my pleasure to be here today and give this presentation, even if it is virtually. I hope that we will be able to interact properly. So the title of my presentation is Engineered Human Cardiac Patches with Physiological Functionality. Let me start by, let me see if I can move. Yeah. Let me start by reviewing something that you already are probably very aware, which is the drug discovery and toxicity testing process. So when a drug, before putting a drug into the market, this drug is discovered through a process of target selection. And the targets, they are going to be tested first in vitro screening. And there will be some drug candidates that are going to be then tested and selected for animal models, and then clinical trials from phase 1 to phase 2 and 3. So overall, this is a process that it's a very long in time. So we are talking about 10 to 15 years process overall. And it's very expensive. So again, we are talking about a total cost of about $2 billion. And overall, it's very, very inefficient process because of the high drug attrition rate. So in the end, overall, 90% of the drugs, they never make it to the clinical trials phases. So from the drugs that they make it to the market, there is another kind of events that they can lead to their withdrawal from the market. And in here, what you have is some of the reasons that can lead to this withdrawal. So these are drugs that are already approved, that they have gone through all the clinical trials, and that they have been put in the clinical practice in humans, and they have to be withdrawn from the clinical practice. And the main reason, as you can see, you have, for instance, hepatotoxicity can be a reason, nephrotoxicity can be a reason, but the main reason for market withdrawal of a drug is cardiotoxicity. And maybe some of them, the best example of drugs that have been withdrawn from the market due to cardiotoxicity reasons is, for instance, defermedine. So when talking about cardiotoxicity, we can have several alterations that might lead to unexpected outcomes that in the end can make a drug be withdrawn from the market. But the most frequent event is cardiac arrhythmias. So in cardiac arrhythmias, basically there are events that they are called toxicity de points, that they can be seen in surface electrocardiograms in patients, and that they are actually due to delayed repolarization, which is usually caused by the blockade of the hERG channels. So let me see. Yeah. So why is this happening into the clinical practice, and it has not been seen before arriving to clinical trials? It is because a lot of the models that they are used, mainly in the in vitro, but also in the animal models, they have a poor predictive capability of the cardiotoxicity of these drugs. So let me just briefly discuss which are these models that are used to check the cardiac safety of a drug. And here what you can see is the most simple model that is used in vitro to check cardiotoxicity. And this is just cell cultures in which at the single cell level, mainly ionic channels, they are checked. This is usually done with non-cardiac cells, and usually only single ion current channels are checked. So it is not a very complete, I would say, model. Then on the other side, we have at the highest level of the integration, the clinical studies. And then in the clinical studies, what the clinicians they see in patients are mainly features that come from electrocardiograms or from Holters, and that they are related to repolarization and arrhythmias. So the problem is that when the drug arrives to this stage, it has been very costly, and there is a risk for individuals. So in between these two extremes of cardiotoxicity testing, we can find several models. So a better improvement on the just single cell ionic channels is the use of cells that are from cardiac origin, so isolated cardiomyocytes. And this can be of different origin, including animals, but also humans. The problem is that cells in humans, they have a short survival, usually in vitro, so primary cardiomyocytes, they cannot be cultured for longer periods of time. And then this was supposed to be alleviated by the use of, for instance, induced pluripotent stem cell-derived cardiomyocytes. But the problem is that these cells usually they have a very immature phenotype and are not completely representative of the physiology of the cardiac cells. On the other hand, we have models of large animals. And in these models, is it possible to test repolarization, contraction, arrhythmias? And usually, large animals are used to test this, such as dogs, goats, pigs, or sheep. The problem in here is that they are costly and labor intensive, and ethical concerns, and also interspecies differences, which make that some of the events leading, for instance, to cardiac arrhythmias are just bypassed if we are using large animal models, and they can be also then seen in humans. So in between these two models, there is a translational gap that aims to be filled with more complex in vitro tissue-like platforms, such as, for instance, engineered heart tissues. So in these little pieces of tissues, it is in principle possible to measure features that are tissue-like, such as, for instance, contraction or action potentials, and they can be derived from various animals, but also from humans, mainly including induced pluripotent stem cell-derived cells. So these models of in vitro of cardiac tissue are our target to be used as in vitro models for drug testing. So which is the strategy, or which features should be reproduced by these cardiac tissue models to be a proper physiological representation of our human heart? We can then turn to the in vivo cardiac tissue environment and see what is in there and what we can try to mimic and integrate in a tissue-derived cardiac model. So in the in vivo cardiac tissue environment, we have the cardiac wall, which is composed by cardiomyocytes, which are the contractile cells, but it's also composed of other cells that are non-cardiomyocytes, and actually they are non-myocyte origin. And it's also very important, the extracellular matrix, which is in this organ, and that it's composed of many extracellular proteins. Among them, the most abundant is collagen. The cardiac environment requires a lot of consumption of nutrients and oxygen, so it's very important to have a proper blood perfusion that can be in vivo. It's done by a proper vasculature, but we will have to kind of mimic in vitro. There is also an electrical pacing and mechanical stretch and contraction because of the function of the pumping function of the heart. And also there is a cell-cell communication and cell matrix communication that provides a set of biochemical factors that are important for the cells to survive and for the proper physiology of the tissue. So our goal in this project was to try to reproduce the native cardiac environment in vitro using multimodal stimulation platforms. So this is a strategy that has already been explored in several publications, mainly to produce what they are called microtissues. And I'm just going to highlight here a couple of strategies in which researchers have used human pluripotent stem cells, cardiomyocytes derived from human pluripotent stem cells, usually mixed with a population of non-myocytes, and seeded on natural-based hydrogels. The preferred hydrogel is collagen. And let me just show you this work in which the researchers have made this sort of bands or rings of gels filled with cardiomyocyte-derived cells, and those are usually placed between two posts, and when they are beating, then they can move these posts, and they can produce this kind of contractile microtissues in which some physiological characteristics, such as, for instance, contraction forces of the heart, can be explored. There is also another strategy that is not based on the maturation upon forced contractile settings, but it's by including electrical stimulation. And I just wanted to highlight this work in which researchers, they have produced these biowires in which there is also this collagen matrix and the cardiomyocytes seated on this gel, and the cardiomyocytes, because of the architecture of this platform, they grow heavily aligned, and they can be electrically stimulated to even maximize this alignment, and then they express more mature features as a tissue contractile phenotype than the ones that they haven't been electrically stimulated. So seeing this works, our goal when we started this project and our working hypothesis was to try to see if we could build not micro tissues, but microscale cardiac constructs, and see if these constructs that will give us valuable information at the tissue level. How this information would be recorded was also like a primary goal in our work. So when we are looking at how cardiac, how the functionality of cardiac tissue constructs is usually evaluated, we have several strategies. The most used is just dissociate the constructs and get single cells, and then, for instance, by patchgram to check the action potential of the single cells and look for specific currents and ions. There is also another strategy, which is usually work with cell monolayers. And in this case, what people do is to try to check the extracellular potential. And for that, they use group of cells or dissociated constructs. And this is done, for instance, using microelectrodes array, what they are called MIAs. And finally, with 3D constructs or larger groups of cells, there is also the possibility of using calcium and voltage-sensitive dyes and then do fast fluorescent imaging. However, we wanted to see if we could add to our constructs a way of evaluating their electrophysiological properties in a way, at a similar way as is it done with human patients, meaning with electrophysiological recording that will mimic and evaluate the tissue level functionality at the macro scale. So to construct this, having seen the literature and then having in mind our goal of building these in vitro models of the cardiac muscle, we hypothesized that if we mix induced pluripotent derived cardiomyocytes with a proper 3D scaffolds, and we provide this construct with the proper instructive signals, we would be able to generate macroscopic cardiac tissue constructs that will resemble the human myocardium, both functionally and structurally. And a good sign that this is true could be if we could use these cardiac tissue constructs to predict drug-induced cardiotoxicity. So the details of the work that I'm going to explain you right now, you can find them in, well, we did a patent application, but you can find it in this publication by Maria Valls. So let me start by then explain you which was the source of the cells that we have decided to use in this approach. So those were induced pluripotent stem cell derived cardiomyocytes that we got in collaboration, working in collaboration with two researchers from the Center of Regenerative Medicine in Barcelona, Olaya Iglesias and Professor Ángel Raya. And those were cells that were derived from biopsies, skin biopsies of patients, and that they were, their status of the phenotype was reverted to be induced pluripotent stem cells. And then with a cocktail of factors, they were differentiated into cardiomyocytes. So this was the, in brief, the differentiation process that was used to have functional cardiomyocytes that were later used to construct our cardiac patches. So cells, they were first seeded in matrigel coated dishes, and then they were going through a differentiation process using several inhibitor molecules along the timeline, just to get cardiomyocytes isolated at day 20 of the culture. So this is, you can, this in these pictures you can see how the growth of these cells is advancing, and then by day 20, what is important is that we have around 72% of cells that are positive for troponin 1 and for myosin heavy chain. And this is how the plate looks like when we stop this differentiation process. And these are the cells that we are going to use for our cardiac models. So we checked a little bit what was the maturation status of these cells indicated by some gene expression. And then we saw that genes related to cardiomyocyte maturation were actually increasing over the cell culture time, but were very far away still from the those found in the adult heart. So we can say that as a single cells in these two-dimensional monolayers, we got cells that were some sort of having a phenotype of cardiomyocytes, but that were not very mature. And even if we saw signals of troponin 1 and connexin, this was not indicating full maturation for these cells. So once we had the cell source, then we decided on the scaffold that we want to use for our cardiac in vitro constructs. And then in this case, we decided to use a commercial collagen-based scaffold. Its a trademark is Matriderm, and this is a scaffold that is usually used for skin wounds. So it's already working in the clinical practice. And the idea is that then we could have a source of a material that was not much variable batch to batch, and that we could get a proper thickness and reliable supply. So these batches, they have a highly porous structure with a pore diameter of around 20-25 nanometers. And they are composed of bundles of collagen that you can see here in orange, and intertwined with some bundles of elastin molecules. That is the second network that you can see here in green. We also check the mechanical properties of these constructs, of this scaffold, and then we saw that they were pretty soft, especially when we checked the mechanical properties at 37 degrees. So we were talking of about 0.05 kilopascals of stiffness. Overall, we thought that these properties, they could guarantee a good cell retention in principle, and a good mass transport inside of the scaffold, matching the soft properties of the extracellular matrix of the heart. So we decided to use this as our scaffolds. Then regarding the other feature that I said was very important for the in vivo cardiac microenvironment, which is the vasculature, we decided to mimic that by using perfusion bioreactors. So in here you have a scheme of the elements of this perfusion bioreactor. So usually you have a medium reservoir and a recirculation or perfusion system, and a bioreactor that might have or not some sensors, and that might include or not several endogenous stimuli, such as mechanical or electrical stimuli. So by including this perfusion in the system, we can guarantee an effective mass transfer in our tissue constructs, because for tissue constructs that are of a thickness higher than 200 microns, it is very difficult to guarantee a proper mass transport only by diffusion. And then we also have a controlled physicochemical environment. On the other hand, the physical stimulation system might support tissue formation and maturation, and the addition of sensors and control systems will serve us to monitor the tissue level, the functionality of the construct at the tissue level. So this is, with these premises, this is the parallel perfusion bioreactor that we designed. So this system is all plug and play, so it's all built into pieces that can go into the autoclave. And then we, every time we have to do an experiment, then we mount a bioreactor with the different branches that we need for every single experiment. It is composed of a medium reservoir and a peristaltic pump to guarantee the perfusion of the system. And then we have this lure manifold in which we can mount several branches of the holding the cell culture chambers. To guarantee that the flow rate is exactly the same on each of these single branches, we added here some flow restrictors that are these microfluidic elements that have a very high resistance, fluidic resistance, in such a way that the flow comes through this port, and then first it fills completely the lure manifold that has a very much lower fluidic resistance, and then it starts to fill individually every of these branches, maintaining the same flow rate in each of the individual ones. In here, we've also added some debubble systems because we want to keep this in culture for several weeks. So this is a very important piece in the system to avoid clogging of the tubing. We also add drug injection pores. And this was important for our particular application because we want to be the system to be used in monitoring drug response. So by this, we can just inject the drug in here and then monitor online the response of the constructs that they were cultured on these culture chambers that you can see in here. Then the system was completed by the gas exchanger. That was this long tube, which is semi-permeable to oxygen, and was used as to increase the oxygenation of the system. So this is how the bioreactor look in the reality. As I said, it's all plug and play pieces. And yeah, so one important thing that we had to thought about was about the flow rate that we were going to use in each of these branches in the bioreactor to guarantee the proper growth of the cardiomyocytes and the cells that we were adding into the culture. So to compute this flow rate, we had to take into account that there is an oxygen consumption. There is an oxygen consumption by the cells within the scaffolds, and there is an oxygen that is dissolved in our medium, and we have a certain number of rates of cells, and these cells, they consume oxygen at a certain rate. So we set up this flow rate that was derived from a few calculations just by first compute, which is the flow rate that happens within the scaffold, taking into account which is the height of the dimensions, of the pores of the scaffolds, which is the scaffold volume and its porosity. And with this value, what we did was to take into account the viscosity of the medium and the average velocity that we computed in here and the pore radius. And with this, we estimated the shear stress. And it was, we matched our shear stress value with this flow rate to be below the shear stress values that have been reported in literature to be detrimental for the cardiomyocyte functionality. So once we had all of this set it up, then we check the chambers, the cell culture chambers, to be added into the bioreactor, and we use two types of cell culture chambers. We use one, this white one, pink one that you see in here, in which there was only perfusion. So the idea is that we had our scaffold seated, placed between two gaskets that were holding it in place, and we just perfused the system in the vertical direction. And then there was another type of chamber, which is this one, the black ones that you see here, in which we have perfusion, but also we added electrical stimulation. So these chambers, they are not available in the market. So what we did is we manufactured and designed this chamber ourselves. And then we added these electrodes that you see in here to provide for the electrical stimulation of the construct. Then when designing this chamber internally, because we wanted to compare it with these other chambers, we designed them to have an equivalent internal geometry in terms of volume. And for that, we also need to define some designing parameters, such as the electrode position, the diameter of the electrodes, and the distance that they had to be apart. To do that, the first thing we did was to perform a set of simulations. And in there, we simulate electrically the silicon gaskets, the presence of the graphite electrodes of different dimensions, the presence of the culture medium with their conductive properties, and the presence of the scaffold and the cells. And running some simulations, we found out that electrodes of 4.6 millimeters in diameter that were separated 1 centimeter and applying an electrical potential of 5 volts were leading to electric field values in the center of the chamber of around 400 and 480 volts meter in a kind of pretty homogeneous manner, and current densities between 575 and 700 amperes square meter, which were values in agreement with the previous publications in the field. Also, the homogeneity of the electric field applied was, to our view, the proper one to be used in these experiments. So we went on and constructed these chambers, and they were manufactured on polypropylene because this is a biocompatible polymer, and this is resistant to ethanol and autoclaving, and it can be machinable in a successful manner. So this is how the chamber looks like. So we have this port for the medium perfusion and these stimulating electrodes. And then we also decided to add here a reference electrode that actually, that actually is contacting the bottom part of the scaffold, which is, this is a cross section of the chamber, which is this pink structure that you see in here. And using this electrode, this reference electrode, and then filling the chamber with the medium and with the construct, we did some measurements of the actual voltage that we were having at the center of the chamber when we were applying this signal of 5 volts peak to peak, 2 millisecond period duration, and 3 hertz of frequency. And we saw that the actual measurements on the chamber were pretty much in agreement with the measurements that we obtained with the simulations. So we then proceed to the experiments of generating, our cardiac human constructs. And then for that, what we did, this is the experimental flow. So we mix cardiomyocytes, we mix cardiomyocytes derived from induced pluripotent stem cells with non-myocyte cells, in this case, human dermal fibroblasts. And then this cell suspension, we just seeded in a one-way perfusion in the seeding loop on the scaffolds that were placed in the cell culture chambers. Then after this seeding, we just take the scaffolds and then we put them into the bioreactor, and then we culture it for 14 days, either under perfusion, these were the control experiments, or with perfusion and electrical stimulation. After 14 days, this is the type of constructs that we got, and then what we check is that if they could beat spontaneously without any stimulation after being 14 days in the bioreactor. And this is what we got. So you can see that the control scaffold is actually beating spontaneously. And this is what we got with the electrostimulated scaffold, that just by the naked eye, you can see that it's beaten much more efficiently and coordinated than the control scaffold. So we went to explore if there was any difference in terms of the cellular features at the cellular level that was explaining this difference in the contractility. And for that, we did some cuttings, and we stained the cross sections obtained for both the control and the electrostimulated scaffolds. And what we observed was that from the cross sections, the cells, they appear to be a little bit more compacted in the electrostimulated scaffold, and the cardiomyocytes, they appear to be a little bit more elongated following the electrical field direction done in the control experiments. We also did some transmission electron microscope experiments, and then we measured the sarcomere width, and we found statistical significance values between the control and the electrostimulated scaffolds. Overall, I have to say that these differences were not that huge to explain the difference in functionality of the constructs, but actually, even if it at the single cell level, they were not the difference were not that huge. In the end, they implied a very different functionality of the overall tissue-like constructs. And to do that, or to visualize that in a better way, what we did was to quantify this contractility of the control scaffolds and of the electrostimulated scaffolds. And this was quantified by the change on the relative area between the contractile and the relaxed state for the control and the electrostimulated scaffolds. And we could see that the difference in the area was higher, much higher for the electrostimulated than for the control scaffolds. We also quantified the fractional area change, meaning not only as in every contractile period versus time, but also over a period of time. And we observed that actually these differences that you see here, they were statistically significant differences. What we also did then was to try to see if these cardiac patches, they were able to respond to, for instance, changes in frequency, as our heart responds when we are, for instance, exercising, and then it increases the heart rate. And then to do that, what we did was to place our scaffolds among two external electrodes of graphite. And then on these external electrodes, we applied voltage, an increasing voltage, and an increasing frequency. And what we obtained was this. So in here, what you can see is the beating scaffold, and these are the two graphite electrodes. And we are now applying 1 Hertz of frequency, and you can see that this contracts overall to with a frequency of 1 Hertz. But now we are increasing progressively the frequency, so 2 Hertz, 3 Hertz, and so on. And you can see that actually the scaffold starts to get into kind of a tachycardia regime and increases the frequency accordingly to the imposed pace until it achieves to it arrives to a point where it cannot follow anymore. And this is the point where we define what is called the maximal capture rate of our scaffolds. And this was quantified, and you can see this parameter. So the excitation threshold was the voltage to start having this contractile behavior on the scaffolds, and the maximum capture rate was the frequency that they could follow. And you could see that these parameters are indicating a higher tissue maturation and better contractility and synchronous contractility in the case of the electrostimulated scaffold than in the case of the control scaffold. We also check how this contractility was happening with regards to the direction of the electric field that was imposed while growing the electrostimulated scaffolds. And then for that, we did some particle velocimeter analysis. And then what we found was that actually there was a higher strain per contraction in the electrostimulated constructs by these strain maps that we can see in here. But not only this, but also that the electrostimulated constructs, they could beat in the direction of the electric field, as you can see here through the quantification of this cosinus 2 theta parameter that correlates the direction of the contractility with the direction of the electrical field applied. So then, as I said at the beginning, it was very important for us to see how we could actually monitor the electrophysiology of the constructs that we were growing. And to do that, we have to remind that we have 3 electrodes in our system. 2 these electrodes that were used for stimulation, but they can be used also to record the signal, the electrical signal of the scaffold, and 1 electrode that can be used and was used as internal reference electrode. So what we did was to use this system to just get the signal that was coming out from the scaffolds. And at the beginning, we got this gray signal that you can see in the background. So it was very noisy. And we were working with a signal processing group at IBEC, and they help us to filter this signal. And then when you filter, what you got is the red signal that we have here in the screen. This was how the electrophysiological recording system was actually looking in real time. So you can see the bioreactor in the incubator, and then the online monitoring system just getting the signal out from the incubator. And this is what we got. So when we monitor this electrophysiological recording, we got something that was reminding very much an electrocardiogram signal. Not that well coordinated for the control scaffolds, but very well resembling an easy electrocardiogram when talking about the electrostimulated scaffolds. Actually, if we quantify the features over integrating this signal over a certain period of time, we are getting these patterns that are representative for the control and the electrostimulated scaffolds, and it resembles very much to the QRS complexes that we are getting in an electrocardiogram in vivo. And this could be also quantified, so we could extract here some parameters such as the relative QRS complex duration, or the relative QT interval, or the relative AQRS. And we saw that all these parameters, they all go in favor of indicating a higher cell synchronous behavior and maturation of our constructs when growing them in the electrostimulated conditions. Finally, we tried to check if the constructs were able to respond to drugs. That was our intended application. And then for that, we took advantage of this drug injection port that we have installed in the bioreactor. And we decided first to check two drugs, that the effect of two drugs that are well known. So the first one is isoproterenol, that is supposed to increase the beating heart rate. And this is what we found, that is actually increasing the heart rate. This is the signal that is coming out from the electrocardiogram in situ after the application of the drug. And then carbachol, which is a drug that is supposed to decrease the heart rate. And this is what we also saw, that is actually decreasing the heart rate. So once we had that, we decided to check if this system was able to predict drug-induced arrhythmias. And then for that, we used a drug which is called sotanol. This drug is an androgenic antagonist and a blocker of the HERG channel. And in patients, it can produce a prolongation of the QT interval that it ends with a phenomenon of arrhythmia or toxar de pointe. So currently, in vitro electrophysiological studies, which are, for instance, performed by the inhibition of this current channel, they cannot predict the arrhythmias that are observed in vivo. So we wanted to check if this was possible with our system. So for that, we just inject the drug in the system. This was the same in the bioreactor. This is the same experiment, but outside of the bioreactor. So just you can see the movie of the effect of the drug in the contractility of the scaffold. And then what you can see is, well, this scaffold was a little bit damaged, but what you can see is that at the very beginning, the scaffold is beating kind of normally. But then you will immediately see that after some period of the application of the drug, now you can see that eventually, from the time to time, it stops beating. And actually, this is the recording of this mechanical contraction. And in here, you can see that there is some contractility, a mechanical contractility that it's not synchronous, but it's arrhythmic. And finally, we just check if these arrhythmic features, we could also record it with the electrocardiogram. And this is what we saw. First of all, we check if we could observe this QT prolongation that it's observed in patients. And we saw that actually over the time, which is these curves are corresponding to the different time periods after the application of the drug, we could see that there is this QT prolongation. And we could also see that after the certain time of drug injection, we could observe some arrhythmic features in the electrocardiograms, and this inversion of the peaks that it's also observed in patients that have arrhythmic behavior. So overall, I hope that I convinced you that I've shown you that we could produce macroscopic human cardiac constructs with tissue-like functionality. This was the strategy in summary. So using induced pluripotent stem cells and fibroblasts, using 3D scaffolds based on collagen and advanced 3D culture systems, and that the cardiac constructs that we were getting from these assays were we were able to obtain tissue-like functionality and features of drug-induced arrhythmia. So they could be considered as a new promising tool in drug cardiotoxicity testing. So just before finishing, I would like to acknowledge the two main contributors from my group to this work, that are Dr. Maria Valls and Dr. Jordi Comellas, our collaborators, Professor Raya and Olaya Iglesias from the CMRV, and Professor Ramon Raya from IBEC, and of course the funding. And I'm looking forward to your questions. Thank you.
All right, Dr. Martinez, thank you so much for speaking with us and joining us today. Your presentation was so interesting, and I can guarantee that we all learned something new or lots of new things. We have a few questions from our audience. Our first question comes from Dr. Alarcon, and he asks, could you think of incorporating immune cells like macrophages in your construct?
Okay, so we haven't done it for this project at all, but we are trying to do that for the other project of the intestine that we have at the moment running in the lab. So in theory, it is possible to introduce them. Although I have to say that our experience with immune cells is that they are kind of difficult to handle, and sometimes the time that they need to grow and their survival is not matching 100% the time that the other cells they need to, for instance, construct end up with a viable tissue. But I have to say that there is a lot of interest in this, and there is something that we are currently investigating, as I said, not for this project, but for the other one in our lab. Yeah.
Perfect. Thank you. We have a few more questions. Dr. Liang is a principal investigator at the University of Ottawa Heart Institute. And first of all, thanks you for a wonderful talk. And is wondering if you have an estimation of the percentage of cells that are in direct contact with the scaffold.
We haven't done any experiment on that, but with the images that we got from the immunostaining, actually it was difficult so we saw the cells completely embedded within the collagen network. But it was actually difficult to know if this collagen, it was because the cells, we had fibroblasts in there, so they would have also produced collagen. So as we had the scaffold growing for 2 or even 3 weeks, it was difficult to know which of this collagen was actually just coming from the cells that we were growing there, and which was originally from the scaffold that we cultured. What I can say is that there was intimate contact of the cells and the extracellular matrix. That was obvious, but I cannot assure 100% that collagen that we saw there was only the collagen of the scaffold.
Perfect, thank you. And then we have actually a 2-part question from David Cortes, a student at the University of Ottawa. I'll ask the first part and give you a chance to answer that one, and then I'll ask the second question. So the first question is, have you considered including mechanical stimulation either before or after the perfusion and electrical stimulation, or in combination to generate an electromechanical stimulation regime?
Yeah, actually we are pursuing that goal in the follow-up of this project in my lab. So I didn't explain here any of the results, but this is our one of our goals, to try to include both electrical and mechanical stimulation to improve the maturation of the tissues that we are getting. Yeah, definitely.
Perfect. And then his second question is, have you performed any experiments using drugs to control cardiac arrhythmias while increasing the frequency of the electrical stimulation to determine the effectiveness of this drug?
No, we haven't done any drug response curve, for instance. I mean, we have done a lot of experiments changing, for instance, drug concentrations and drug times, but we haven't set it up a proper dose response curve. And the reason is that these scaffolds, they consume a lot of cells. So you really need a huge amount of cells. I think you saw it, like 3.5 million of cardiomyocytes derived from induced pluripotent stem cells. So, and then you have to keep them in culture for a long period of time. So this means that all these experiments are pretty expensive. And so you really need, and time consuming. So you really need somebody that is fully committed to the experiments, and you really need quite a huge amount of money to do all of these experiments in a proper manner. So we didn't have the chance so far to do that. Yeah.
Thank you. And we have two more questions. Our next question comes from Hallie at the University of Ottawa. She asks, were there any differences between the control and the electrostimulated scaffold in terms of the mechanical properties, specifically the elastic modulus, after being in the bioreactor? Is the electrostimulated scaffold similar to the physiological elasticity of cardiac tissue?
We haven't checked that, actually. That would be very interesting to check, but we haven't done it. So the reason is that for logistic purposes, once we had the experiments and then we record all the data within the bioreactor, then we usually took out the scaffold and then we observed the mechanical contractility, and we kind of repeated the same kind of experiments to have the correlation between the mechanical contractility and the ECG signals that we got from the bioreactor, because there you can see anything. And then once you arrive to the point that you've completed the whole process, then later on we wanted to fix the scaffold to be able to check for immunostainings. So within this process, I mean, logistically for us, it wasn't really very convenient to take up, because you also need quite a few replicas, to take these replicas and go and measure them in the DMA system. But of course, this is interesting, and I'm sure there will be some differences. I'm not sure how important they will be, but the cells are placed and arranged in a very different manner. So this should involve some differences. Yeah.
Very interesting. We have a few more questions. They keep coming in, which is good. Our next question is from Delphine Gordon, and who says, very nice talk, and asks, what is the average cell density in your scaffolds? Are they homogeneously distributed?
Yeah, just maybe let me go back to the slide where I had this. Okay, so in the electrostimulated scaffolds, they are not homogeneously distributed. They are actually concentrated in this region that is where the electrical field is maximized. So the electrodes, they will be placed just one here and one here, and the cells, they tend to accumulate in this region, leaving this region and this region free or almost free from cells. And this can be also seen, yeah, in these cross sections. Green here is for the cardiac troponin. So you have cells, we have cells in the control, for instance, all around, but the cardiomyocytes are accumulated in a certain regions. And in the electrostimulated scaffolds, the scaffold is much more compact, if you want, maybe because of the contractility, so which is higher. And we could see this accumulation of the cells in the central part of the scaffold, corresponding to the place where the electrodes were located. Cell density, I mean, this is how it looked like. So we didn't measure that, but they were really fully packed. So it was a very compact tissue-like piece. So when you get it out, it's like something very similar to a meat slice, if you want.
Perfect, thank you. And then I believe this is our last question from Gabrielle, a student at Carleton. And she asks, how accurately can microtissues detect new problems with untested drugs?
I didn't understand could you repeat the question?
Yeah, sorry. She asks, how accurately can the microtissues detect new problems with untested drugs?
Okay. So I have to say that there is a lot of efforts in testing this. So there are not microtissues have not been approved as a valid in vitro testing method so far. So, but still, even if they are not approved, what I can say is that people and especially pharma industries, they are interested in microtissues, and they are interested because they can get some information that they cannot get using other in vitro systems. So this means that even if they are not at the point of being used as validated assays, although there are some efforts towards that part, because mainly the main problem is throughput, so you really need quite a huge throughput to get a proper validation method. But even if this is a challenge, there is interest in using them. And this is because of the information that they can be combined, maybe despite of the throughput, it can be combined with the other systems that are already validated. And this can produce altogether a prediction, which will help to decide in moving forward or not with that drug candidate. So that's a little bit the idea and the status at this moment.
Sorry, there is one more question. And I apologize if I say your name wrong, from Madhya Hijargujani. Asks, why did you not use iPSC tube cultured on the scaffold and let them differentiate to cardiomyocyte?
Yeah, we tried that and it didn't work. So we don't know why, but the cardiomyocytes that were not differentiated the IPS that were not differentiated to cardiomyocytes, they were not able to. Sorry, we have a spontaneous here. They were not able to grow, so they died after a few days in culture. We still don't know why, but these experiments they didn't work so far.
Perfect. I don't believe that there are any more questions. On behalf of the Canadian Biomaterials Society Ottawa Student Chapter, thank you so much for tuning in this morning, and thank you, Dr. Martinez. And everyone, please stay safe and take care. Thank you again.
Thank you. Thank you very much. Bye-bye, everyone.
Bye.